In this video we're going to learn about the deoxyribonucleic acid, commonly known as DNA, which is one of the most important questions. The structure of the DNA has been asked 4 times, and the semi-conservative method of replication of the DNA, it is asked 8 times in the past 23 years, so it is a very important question. So before understanding about the structure of DNA, we need to deal with some minor terms in order to understand the structure of DNA. So first of all, we need to know what is a genetic material. So basically, genetic material, it is the substance or the material that gets transformed from one generation to the other generation, and it expresses the parental characteristics in the offsprings. So it gets transmitted from one generation to another generation to express the parental characteristics. Now in human beings, in the living organisms as well, almost in all living organisms, the basic genetic material is DNA and RNA. It means DNA and RNA, they perform the work of getting transmitted from the generation to the next generation, and it transmits the parental characteristics in the offsprings. So after this DNA, we hear a term called gene very often. So actually, what is a gene? So gene, it is the unit of the DNA, to be precise, the functional unit of the DNA. Actually, this gene, it is a part of the DNA, or we can call it as a portion of the DNA which produces a particular trait. Now trait means it produces a particular hereditary pattern. It means one gene, it is responsible for the transmission of one type of characteristics to the offsprings. It can be either height, color, anything. Any one of the hereditary characteristics, it is determined by the gene. So gene is known as the functional unit of the DNA. So basically, gene, it is a portion of the DNA only. So the DNA, it is described as an information tape which encodes the messages for all the vital activities and transmits it from one generation to the next. Okay, that was just a glance of the DNA. Now we're going in detail. So actually, what is a DNA and where is it present? We need to understand this first. So DNA, it is a type of nucleic acid. Okay, so basically DNA and RNA both are the nucleic acids, they are the genetic materials. So nucleic acids, they are the most essential molecules of life. These are very complex long chain compounds which are even complex and larger than proteins. They contain carbon, they contain oxygen, they contain hydrogen, and chiefly nitrogen, phosphorus. There are 2 types, DNA and RNA. So basically, nucleic acids, they are the compounds or they are the polymers. Polymers means that they are formed of combination of small units which are known as monomers. These monomers combine and they form polymer. This process is known as polymerization. So basically, these small monomers of the nucleic acids are the nucleotides. Okay, now don't be confused with nucleoside and nucleotide. I'll tell you what is the difference between nucleoside and nucleotide. For now, nucleic acid, basically it is the compound which is a polymer of nucleotide. It means many thousands and millions of the nucleotides, they combine and they form a polymer which is known as nucleic acid. So they are of 2 types, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Primarily, nucleic acids serve as the transmitters of the genetic material. It was first discovered by a scientist known as Johann Friedrich Metzger, okay, in 1868 actually. So that was just a brief history about it. Now I'm telling you in detail about the nucleotide, what is a nucleotide, and then you'll understand the DNA. So nucleotide, it is a compound which is formed of 3 components: a base, a sugar, and phosphate group. So what are these things? I'm going to tell you. First of all, see, a term nucleotide, it refers to base plus sugar plus phosphate group. So it is just a combination of a base, a sugar, and a phosphate group. Now the components of the nucleotides, in precise, it is pentose sugar, a sugar which is the pentose sugar, base which is the nitrogenous base, and the phosphate group means phosphoric acid. So basically a nucleotide, it is formed of a pentose sugar, a nitrogen base, not a nitrogen base, there are many nitrogen bases, and then phosphoric acid. Let's go in detail about this. And one thing, this structure of nucleotide is never gonna ask you in grade 12. In 11 it might be asked, but in grade 12 it is not asked usually. I'm just telling you for your information, just so that you can understand the DNA better. Now pentose sugar, it means the sugars which are formed of the 5 carbon monosaccharides. Now if you see detail about the sugar, the basic unit of sugar is monosaccharides. Okay, this is the basic unit of sugar. Sugar is a polymer as well, which is formed of monomers known as saccharides or monosaccharides. Mono means one, so it is formed of monomers called monosaccharides. Now these monosaccharides which contain 5 carbon, it is known as pentose sugar. If you write it structurally, the formula, it will be C5H10O5. Now let's not go in much detail about this, just I want to tell you is pentose sugar, it is a 5 carbon monosaccharide. They are of 2 types, ribose sugar which is present in the RNA and deoxyribose sugar. Okay, and deoxyribose sugar, it is present in the DNA. By name it is clear. Now these both, ribose and deoxyribose, they basically differ in structure at the second carbon. If you see the structure of the ribose and the deoxyribose sugar, it will be somewhere like this. Now you do not need to remember the structure, I am just telling you for information. Now this is ribose and this is deoxyribose sugar. They differ in the structure in the second carbon. If you count from here, 1, 2, 3, 4 and 5, 5 carbons are here. Okay, 1, 2, 3, 4 and 5, so 5 carbons are there, and in the second carbon the ribose contains OH, but the deoxyribose only contains H. So this is the basic difference between deoxyribose and ribose sugar. And that was all about the pentose sugar. The thing we just need to remember is that there are 2 types of pentose sugar, ribose and deoxyribose. Ribose in RNA and deoxyribose in DNA. Then we have the nitrogen bases, which are the most important part of a DNA. Now we'll study about this nitrogen bases in detail. Now nitrogen bases, they are of 2 types. There are 2 categories of nitrogen bases. We are not saying there are 2 types of nitrogen bases. The types are 5 types. We have 2 categories of the nitrogen bases. First we have purines, another we have pyrimidines. Okay, purines and pyrimidines. Now the difference between these 2 categories, when we draw the structure, the chemical structure, we'll find that purines are 2-ring structure. Like suppose this is the structure of purine, any purine, then pyrimidines, it will be 1 ring in structure. So purines basically they are 2 ring in structure, and the pyrimidines they are 1 ring in structure. Now these rings are of the carbon nitrogen rings. Somewhere we have carbon, the nitrogen, carbon nitrogen, somewhere like this carbon nitrogen ring are these purines and pyrimidines. And now here is the major thing about the purines and pyrimidines. Purines, they are of 2 types. We can even call it the examples of the purines. First one is adenine, which is represented by a capital letter A, and then guanine. Now you need to remember this. Purines are adenine and guanine. Okay, you can remember A, G. They are adenine and guanine. Pyrimidines are of 3 types. So first type of pyrimidine, it is the cytosine, which is represented by a capital letter C. Cytosine. Then we have a thymine, or we can call it thymine. Anything, pronunciation is different. Now we have uracil. Now uracil is represented by U. So adenine, guanine, cytosine, thymine, and the uracil. So these are the 5 nitrogen bases. Now these 5 nitrogen bases are must to remember when we are reading about the DNA, because it plays an important role in the DNA. Basically it is the most important thing in the DNA, which determines the character of each and every individual. So adenine, guanine, purines, cytosine, thymine and uracil are pyrimidines. Now what is the difference between them? As I told you, the difference is that they are 2 ring in structure and they are 1 ring in structure. So nitrogen bases, we knew the types, but what is this types about? Now basically in a DNA 4 nitrogen bases are present. Even in RNA 4 nitrogen bases are present. Now the difference comes here between thymine and uracil. These 3 are present in both of them. It means adenine, guanine and cytosine. This will be present in both RNA and DNA. But the difference is that DNA contains thymine and RNA contains uracil. Now if you go in detail about the thymine and uracil, you will find that thymine is also a type of uracil. It is just known as the methylated uracil. It means in structure they just differ in a CH3 group, in a methyl group. Thymine contains methyl group and uracil does not contain. So thymine is also known as methylated uracil. Okay, to the point, DNA contains A, G, C and T and RNA contains A, G, C and U. That is the difference between DNA and RNA. They differ in thymine and uracil, but they are formed in the form of complementary bases. They are present in the form of complementary bases. Now what it means is that a purine base, it is always attached to a pyrimidine base. Like if you see the structure of DNA, we'll find that the adenine, it will be attached to the thymine always. It means a purine is always attached to a pyrimidine. It is present in the form of complementary bases. One is complementary to another. It means one is present with another. So a purine is always present with pyrimidine in a bond. Now that bond may be double bond, maybe triple bond. So adenine is always present with thymine, okay, and they are present with double bond, 2 hydrogen bonds. And cytosine, it combines with guanine. It means guanine, it combines with cytosine. So they are present in triple bond. Now it can also be uracil in presence of the in presence of thymine. It can be AU in the presence of thymine, uracil in RNA, but that's the case of RNA. We are dealing about the DNA. So in DNA, if we see, A and T are present as double bond, C and G are presented as triple bond. Now again I'm telling you from the beginning, nitrogen bases are of 2 categories, purines and pyrimidines. Purines are adenine and guanine, pyrimidines are cytosine, thymine and uracil. In a DNA, adenine, guanine, cytosine and thymine is present. In an RNA, adenine, guanine, cytosine and uracil is present, but they are present in the form of complementary bases. It means bonded with one another. So adenine is found with thymine, bonded with 2 hydrogen bonds. Guanine is found with cytosine, bonded with 3 hydrogen bonds. You can remember ATCG, okay? So that's the only way we need to remember AT and CG. They're by double bond, triple bond. And during this type of purine pyrimidine bonding pairing, the total number of pyrimidines is always equal to the total number of purines. It means if you see the total number of purines in DNA, it means A plus G, the total number of purines is always equal to the total number of pyrimidines. It means C plus T. This number is always equal because A is always equal to T and G is always equal to C. So A plus G is always equal to T plus C, and this rule is known as Chargaff's rule. Okay, this is an important rule. We can even write about it in the structure of DNA. It is known as Chargaff's rule. But remember one thing, the number of adenine is not equal to the number of cytosine. Okay, it means the adenine is equal to thymine, the purine is equal to pyrimidine, but it is not necessary that adenine will be equal to the cytosine or adenine will be equal to guanine. It is not necessary. Like 2 types of purines are there, the number of adenine can be 5, the number of guanine can be 7. It means they both can differ. So remember, adenine is not equal to guanine and cytosine is not equal to thymine. The equal one is A is equal to T, G is equal to C. Don't get confused. Purines is equal to pyrimidines. So that's about the nitrogen bases. And finally we have the last one, which is the phosphate group. You see, it was the phosphoric acid. Now there is nothing much to tell about the phosphoric acid. It is just a phosphate group which combines the 2 nucleotides. Now as I said, nucleotides, they are the monomers. So suppose this is one nucleotide and this is another nucleotide. So these both nucleotides are combined by a bond. Okay, this bond is known as phosphodiester bond. Phosphodiester bond. So it is a bond of the phosphoric acid. So this phosphoric acid, it combines two nucleotides together by a bond called phosphodiester bond. So we can also call it is a backbone of the nucleic acids, because it is combining the two nucleotides and then the entire nucleic acid is formed. So that's all about the three components of the nucleotides. Now yeah, I was going to tell you about the nucleoside. So as I said, nucleotide, it is the base pentose sugar plus nitrogen bases plus phosphoric acid is a nucleotide. Now there is only one thing difference in the nucleoside. Nucleoside, where S is present in place of T. Nucleoside, it is just pentose sugar plus nitrogen base. Pentose sugar plus nitrogen base. It means if we do not count the phosphoric acid, if we only count the sugar and the base, only the pentose sugar and the nitrogen base, we have to call it a nucleoside. And if we are counting the phosphoric acid, it means the phosphate group, so we need to call it nucleotides. That's all about the nucleoside and nucleotide. And then we jump on our major topic, which is the DNA. So till now I just gave you the information to understand the DNA. So now here the questions of the examinations begin. The first question will be asked, the structure of the DNA. So here I'm going to tell you about the structure of the DNA now. So DNA, basically it is present in the nucleus. If you see the location of the nucleus, we'll find that the DNA, it is present in the nucleus, in the mitochondria, and even in the plastids. So these are the three major places where we can find the DNA of an organism: the nucleus, the mitochondria, or the plastids. In the nucleus, it is present in the chromosomes. The chromosomes are basically made up of the DNA, which is covered by a protein called histone protein. So that was the location of the DNA. Now if you see the history of the DNA, DNA was first discovered in 1869 by a scientist called Frederick Mister. He discovered the DNA in 1869 for the first time. But the Nobel Prize for the demonstration of the double helical model of the DNA, it means the structure of the DNA, was given to two scientists, James Watson and Francis Crick. Now remember the name, it is a very important question which is asked in 1 marks. Who was awarded as the Nobel Prize for giving the double helical structure of the DNA? So the first name is James Watson and another is Francis Crick. They were in 1953, they were awarded as the Nobel Prize, with the Nobel Prize, because they gave the double helical structure of the DNA. Now what is the double helical structure of the DNA? Basically it is the structure of the DNA which says the DNA is in the form of a double helix. Okay, now I'm going to tell you about it in detail. So we are going for the structure of DNA now. So Watson and Crick model of DNA, it possesses the following features. We'll write point wise, so it will be easier for us to understand. Even in examination you can write it point wise. So first of all we need to see that DNA, it is a double helix. Now what is meant by a double helix? It means it consists of two polydeoxyribonucleotide chains. Now again I'm repeating, it consists of two polydeoxyribonucleotide chains. Now what is meant by this word polydeoxyribonucleotide? We know that DNA is a nucleic acid. Nucleic acid is made up of nucleotide. Okay. Now basically nucleotides are of two types: deoxyribonucleotide, ribonucleotide. Ribonucleotide is in RNA, deoxyribonucleotide is in DNA. So it is deoxyribonucleotide. But we know that nucleotide is just a monomer of the nucleic acid. So deoxyribonucleotide, it is just a monomer of the DNA. For it to form a DNA, it should be in large quantity, it should be the polymer. So it is known as polydeoxyribonucleotide, because it is just a monomer and this becomes a polymer, and that polymer is known as DNA, a nucleic acid DNA. So it is a double helical structure and it consists of two polydeoxyribonucleotide chains. It means one chain is there, okay, and with it another chain is there. So it has two chains of polydeoxyribonucleotide. Basically it is a nucleotide. Now they are twisted around each other. Double helical means these two chains, they are twisted around each other. They are not present parallelly, they are present with twisted among each other. And the axis is common. It means the axis, this is the axis, the central portion, it is the common one, and they're twisted. And we can see it is somewhat spirally twisted. This two spiral strands of the DNA, the two spiral arms of the DNA, we can collectively call it as the DNA duplex. So again, the first point I'm telling you again, a DNA is a double helical structure. It consists of two polydeoxyribonucleotide chains which are twisted among each other along a common axis. Okay, it is spirally twisted, and the two spiral strands of the DNA, they are collectively known as DNA duplex. Now that was the first point. We have the second point, which is one of the most important points in this. Each nucleotide, each deoxyribonucleotide, it is made up of a sugar plus a phosphate group plus nitrogen bases, and the nitrogen bases which are present, they will be our another point. Okay, so we are making as much points as are possible, so we do not write everything in a single point. We'll make as much point as possible. So first of all, double helix, another one, each nucleotide, it is formed of sugar plus phosphate plus nitrogen bases, and yeah, two nucleotides, they are attached to each other by phosphodiester bonds. We know this as we read in nucleotide. They are linked together by phosphodiester bonds to form a polydeoxyribonucleotide chain. Okay, now we're going for the third point. Let's go to a new page. So we had the first point, we had the second point, now we have the third point. Now we said that the DNA, it consists of two strands which are spiral twisted among each other, but the factual thing is these two strands of the DNA, they are anti-parallel. Now what is meant by anti-parallel? If we see this strand, okay, let's make the figure here, the rough figure of the DNA. If this is one strand of the DNA and this is another strand of the DNA, so if we see one strand is running in this direction, suppose this strand is running in this direction, then this strand will be running in opposite direction to this. We denote it by numbers which are 3 prime to 5 prime. Okay, we denote it like this: 3 prime to 5 prime. This 3 prime to 5 prime, this is the first strand, and the second strand will be 5 prime to 3 prime. Okay, see, 3 prime to 5 prime, and this will be 5 prime to 3 prime. Suppose this direction is 5 prime, this direction is 3 prime. If one is running from 3 prime to 5 prime, then other will be running from 5 prime to 3 prime. Okay, this is just a symbolic notation that the two strands are running in opposite direction. Now what is meant when I say they are running in opposite direction and why it is running in opposite direction, we will be learning about it in the DNA replication. You will understand about this in detail in DNA replication, which is our next topic. For now, just understand that the two strands of the DNA, they are anti-parallel. If one is running in 3 prime to 5 prime direction, then the other strand will be running in 5 prime to 3 prime direction. So this was just a complex point of the structure of DNA. The other points are very simple and very easy. The fourth point we have, it is about the width of the DNA. So if we see the width of the DNA, the width of the two strands, it means the distance between the two strands, it is exactly same in all central portions, and this diameter is of 20 Armstrong. So basically when we see this whole diameter, the width of the DNA is 20 Armstrong. Each DNA has a width of 20 Armstrong. Okay, so width is 20 Armstrong. Now Armstrong is a unit which is expressed in terms of 10 to the power minus 10 meters. Okay, so 20 Armstrong. Now we have the fifth point, which is the length of the DNA. If we see the distance of one turn of the DNA, suppose this is one turn of the DNA. Okay, now see, this is not the one turn of the DNA. This complete turn is the one turn of the DNA. Again, I'm making the figure of this is one helix, this is another, sorry, one strand, another strand. So from here it goes and it ends here, and another one, it goes and ends here. So this one complete turn, this one complete turn of the DNA, it is of 34 Armstrong. Okay, so one turn of the DNA is 34 Armstrong. Now remember, one turn consists of 5 nitrogen bases. There are nitrogen bases on the inner side of this DNA. Okay, 5 here, 5 here. So basically there are 10 nitrogen bases in one turn. So if there are 10 nitrogen bases in one turn, then a distance between one nitrogen base will be 34 divided by 10, which is 3.4 Armstrong. So again I'm telling this point. First of all, the width one, the width of the DNA, the diameter of the DNA, the distance between the two strands, we can call it as 20 Armstrong. Okay, angstrom is the unit which is in the form of 10 to the minus 10 meter. Now one turn of the DNA, basically this is one turn of the DNA. So one turn of the DNA, it is of 34 Armstrong. Since one turn of the DNA, it consists of 10 bases, 10 nitrogen bases. So the distance between each nitrogen base is 34 divided by 10, 3.4 Armstrong. Okay, so that was our fourth and fifth point. Now we have the sixth point, which is about the nitrogen bases. Now we can write about the nitrogen bases. They are the nitrogen bases present in the DNA. They are adenine, which is paired with thymine with 2 hydrogen bonds, and another is guanine, which is paired with the cytosine with 3 hydrogen bonds. We can write about this. This can form our sixth point. And then we have the seventh point, and here we can write about the Chargaff's rule. As I said, we can even write about the Chargaff's rule in the structure of the DNA. Now if you forgot the Chargaff's rule, I'll tell you again. Remember, the purine is always equal to pyrimidine. So the number of A plus G will be always equal to T plus E. The total number of purine is always equal to the total number of pyrimidine. So that is our seventh point. And even these points will be enough. Still I'm telling you about the eighth point, which is about the minor groove and the major groove. So first of all, I'll draw the structure of the DNA here, the figure we need to draw, then I'll tell you about the point 8, or else you'll get confused about it. So after writing all these 7 points, okay, I'll repeat all the 7 points again. So first point was the double helical structure of the DNA. The DNA is a double helix structure. It means it consists of 2 polydeoxyribonucleotide chains. If you forgot the term, you can just write it consists of 2 nucleotide chains, okay, which are spirally coiled among each other, twisted among each other along a common axis, and these both strands are together known as DNA duplex. So that was the first point. Second point, each nucleotide, it is made up of a sugar, a phosphate, and a nitrogen base. And many such nucleotides, they are linked by phosphodiester bonds to form a polynucleotide chain. Now these two strands, the third point, these two strands of the DNA, they are anti-parallel. It means if one runs in the direction 3' to 5', another will run in direction 5' to 3'. Then we have the width of the DNA duplex, or we can call the diameter or the distance between the two strands, it is 20 angstrom. The distance between one complete turn, it is 34 angstrom. The distance of one turn is 34 angstrom, and each turn, it consists of 10 base pairs. So the distance between each base pair is 3.4 angstrom. Now the base pairs are in such form: A pairs with T double bond, and G pairs with C triple bond. Then we have the Chargaff's rule. The purine is always equal to pyrimidine. Okay, now first of all, as I said, the structure, and then I'll tell you about the 8th point. So if we draw the structure, the detail, not this rough structure, a clear one, we'll first draw our DNA strand here. Okay, and now give a major distance. Remember, draw this and give a huge distance between them, huge distance, and then okay, this much will be enough. So make it double. So now here, see, we'll draw another strand. So while drawing another strand, remember here there should be huge distance between them. So just draw it till here and then make it like this, till here, and twist it. Now here there should be a huge distance between them, huge gap between them. So now see, I drew the structure. First of all, I drew one strand which is drawn in such shape, okay, a huge distance between them. Then I drew another which is just opposite of the structure of this. Okay, but remember to make a huge gap here. You draw till here and then just go parallel with it. Okay, we just go parallel with the first strand. You draw till here and then just go parallel with the first strand. See, parallel with it like this. and here parallel, so that the huge distance between them is maintained. Now why I am considering this huge distance much? Because it is our 8th point. Now this huge distance is known as a major groove, and these small distances, they are known as minor grooves. Okay, so they are major groove and minor grooves. There are, so 8th point is that a DNA duplex, it consists of major grooves as well as minor grooves. So there are two grooves in the surface of the DNA molecule. One is wide and deeper one, which is the major groove, and another is narrow and shallower one, which is the minor groove. So that was about the points of the DNA, and now the structure of the DNA. Now we have the figure. We need to make the figure very clearly. So this was just the structure. Now remember that these outside one, they are the sugar phosphate backbone. Sugar phosphate backbone. It means it consists of the pentose sugar, it consists of the phosphate group which are linking the two nucleotides. So it consists of sugar phosphate backbone. Now sugar and phosphate group are outside, so remaining one, the nitrogen bases, they are inside. The three components, the nitrogen bases, the pentose sugar and the phosphate, among them sugar and the phosphate are outside and nitrogen bases are inside. Now each turn should consist of 5 nitrogen bases. You can make it randomly, A with T, C with G, A with T, C with G. Okay, but remember, if you are making, you need to consider the bonds. So in this way you can make the inside one. And now if you remember the figure clearly, first of all we'll complete the detail of the figure. We know the width is 20 angstrom. Now the width of one turn is 34 angstrom. So the width between two nitrogen base is 3.4 angstrom. Okay, now I was saying that if you remember the figure completely, then you can write the points of the DNA in your own words, and you do not need to mug up the points of the structure of DNA. You can just write along with the figure. As the figure is showing, there are 2 helix, it means 2 strands in the DNA, and you can write that it is made up of sugar phosphate backbone and then nitrogen bases on the inner side. Then you can go that they both are anti-parallel, and you can write about the distance by remembering figure, and then you can write about the pairing. So the figure is very important. So that was the Watson and Crick model. Now I'll tell you about one more figure, which is the double helical structure of the DNA, which was again given by Watson and Crick, but it is much detailed figure. This is the structure of the DNA. Now see, there is one more structure. Now this, you need to draw this figure, and the figure I'm going to tell you, it is not necessary, it is just the detail. So as we saw that there are 3 components, the phosphate group, the nitrogen bases and the pentose sugar. So how are they present actually? Here we are showing that if you see, the bonding is present there between the purines and pyrimidines. Inside this, the bonding is present. So let us draw it rectangularly. The bonding is present, and then even the T and A, it can be present randomly. You can draw it randomly, anything. You can even draw now the C and G. You can draw anything, but the triple bond should be mentioned. So now what happens is that this is just the bonding. Now on the outer surface, the pentose sugar is present. This is the bonding on the inner surface, and on the outer surface, the bonding of the pentose sugar is present. Now we know pentose sugar, as I said, it is 5 carbon monosaccharide. So we'll draw it like a pentagon because it is 5 carbon monosaccharide. This form, sorry, this is a hexagon, somewhat like this, 5 carbon monosaccharide. So we have to draw somewhat pentagonal structure. So you can draw it like this. As you wish, just the pentagon should be there. Okay, so this is our pentose sugar. Now even on this side you can draw, I'm just telling you for information, even you can draw this figure in the examination without drawing that DNA model. Any figure will be worth it. Now this is the pentose sugar, these are the nitrogen bases, and this is a nucleoside. Now here you will get clearly about the nucleotide and nucleoside. See, only the pentose sugar and the nitrogen bases are present, no phosphoric acid right now, no phosphate group right now. So this is one nucleoside, this is another nucleoside, another nucleoside. So now when we join these both with the help of a phosphodiester bond, see, phosphodiester bond, we are considering it they are joining the two pentose sugar because it is present on the outer side sugar phosphate backbone, so they are present outside. We'll draw circular structure. So this is our phosphate group, so let us PG, we can tell it, okay, phosphate group. So they are just joining the two nucleosides and now it becomes a nucleotide. So this is a nucleotide, now this is another nucleotide, now this is another nucleotide, which are joined with phosphodiester bonds. So these both are ester bonds. Now since two ester bonds are there, it is known as diester bond. So this is the reason why it is known as phosphodiester bond. So the phosphate group which are joining the two nucleosides with phosphodiester bond. Now this is about the structure of the DNA. With this we complete the structure of the DNA, and one thing remaining, see, this is one strand, this is another strand. If this strand is running in 3' to 5' direction, this strand will be running in 5' to 3' direction, the opposite directions. Okay, so with this we complete the structure of DNA. You can make the either of the figures. I think this figure will be much easier. Still, it's your wish, you can make any of the figures. And now with this we complete the structure of the DNA. The thing you need to write in the examination is pretty simple, just a figure and then the 7 or 8 points as you wish, and yeah, you'll be getting 7.5 marks for this question as well, or 8 marks. So that was all about the structure of the DNA. Now in the next video we're going to learn about the DNA replication, which is another important topic of this chapter. Till then, take care, goodbye.